Okay, so let's talk about kind of a little bit more nuts and bolts details. Okay, so you kind of get the big picture what photosynthesis is about. Let's talk about some of the details. All right, so light reaction. Light reaction is a reaction that obviously takes place when there is light. So you need to know a couple things about light. Light travels in what's called waves. Waves have a length and a height. So I'll use the analogy of going to the beach. Um, Wavelength also has to do with the thing called frequency. So, for example, you go to the beach and the waves hit you every 4 seconds. All right, not a big deal. So you're standing there, waist deep water, wave hits you. Let's say the wave is, maybe a foot high, hits you in the chest and goes by. Next one comes in, hits you in the chest and goes by. The length of the waves is how often they hit you. So the frequency would be every 4 seconds is the length of the wave. So we could say that the wave has a 4-second frequency. The waves become stronger as more waves hit you. So, for example, let's say in the course of, we'll use a minute worth of time. Let's make the numbers easy. If the wave frequency was every 10 seconds, you'd be hit by 6 waves in a 1-minute period. If I increase the frequency of the wave, so how often the wave hits you, it becomes harder to stand up in the water. Why? Because there's more energy hitting you. Let's say the waves have a 1 second duration, or 1 second between waves. You're getting hit 60 times as opposed to 6 times in the course of that same 1 minute. So the wavelength is the length of time or how often the waves hit. Okay. Another thing that you can talk about with waves is the intensity of the waves. So let's say you go to the beach on a nice calm day, no wind. The waves might be pretty small. Right? So you can have waves that maybe hit you every 10 seconds. So that would be the length of the wave or duration of the wave, but they're relatively small. You go back the next day after a storm, and let's say the waves are still hitting you once every 10 seconds, but instead of being, a foot high, the waves are 3 feet high. So each wave that hits you is carrying more energy. So waves they have a length and they have a height or an amplitude. Okay, think where we can put this in kind of perspective would be light bulbs. So when you look at a light bulb, the light bulbs have numbers on them. Right? The numbers are rated in wattage. The wattage is not the wavelength. That's the intensity or the height of the wave. So if you take a 2-watt bulb, a 4-watt bulb, an 8-watt bulb, as the wattage goes up, the height of the wave, the strength of the wave goes up. So a 200-watt bulb would have lots of energy, and you can see this in class. Wavelength is really the colors. So every length of a wave is going to be a different color, and we'll talk about this electromagnetic spectrum. So here's the spectrum itself. So we have this thing called visible light, and the waves go from shorter to longer. Okay, the shorter the waves, the more often they hit you. Okay, the longer the waves, the longer period between the waves, the less energy that wave is going to have. So shorter waves have the most energy. All right, longer waves have less energy, and they go Roy G. Biv of the colors: red, orange, yellow, green, blue, violet, indigo. All right, they go in that order. So here's our red, orange, yellow, green, blue, violet, indigo. So the Roy G. Biv. Okay, the red waves have the least amount of energy. Purple have the highest amount of energy. Notice they're closer to UV. UV stands for ultraviolet. Okay, these waves here from gamma, X, UV, infra, micro, radio, they get progressively longer. The ones that you're probably most familiar with are X-rays, UV, which would be from the sun, all right, infrared, which is heat, microwaves, those things you have at your house, and radio waves if you're listening to the radio, not XM radio, but if you're listening to AM or FM radio in the car, those would be radio waves. The only ones that we can actually see are visible light. You can occasionally see infrared, but that's we'll kind of get into that conversation in class. X-rays we can't see. We have to develop them to see those. UV are once again invisible rays that actually cause cancer. So most of our cancer-causing things are going to be down in this end. We don't see as many cancer-causing waves up here, although we Okay, back at it. So, yeah, chlorophyll more like borophyll. Little Happy Gilmore reference. Um, anyway, so chlorophyll A are so here's a graph here of the different chlorophyll colors. This is it's a spectrophotometer. So what you can do is you can take we have this little device that it measures color. So if you've ever been to like Home Depot or Lowe's and you want a certain color paint, you bring in a little chip. They can measure the color of that paint using different wavelengths. So what they do is they separate the colors, bounce the color at whatever the object is. Whatever color bounces back, they know that's the color of that particular object. Because, for example, chlorophyll A, okay, is green. So if you look down here, notice it shows you absorbance of the light by chloroplast pigments. So chlorophyll A is going to absorb colors from 380 all the way through 400. It's going to peak somewhere about 425 or so, and then it drastically goes down. When you get closer to 500, which is the wavelength of green, notice that it virtually absorbs zero light. All right, so if it's not absorbing light, it's reflecting light, and it doesn't really absorb very well at all in this green spectrum. It doesn't. It's not till about it to red again it starts to absorb. So what happens is chlorophyll A absorbs or does its best work. Has two peaks, somewhere around 425 and somewhere about 680. Okay. Chlorophyll B, which is a second version of chlorophyll, and you're going to remember that chlorophyll. We talked about organisms basically being big sacs of proteins. Chlorophyll A is a combination of proteins and fats that are made by plants. Okay, mostly proteins, but they do have some fats mixed in there. So here's chlorophyll B. Chlorophyll B, once again, starts working from 400 all the way up, and it peaks somewhere about 480 or so, 475 maybe, and then it drastically drops off again. Does not absorb very well between that green and yellow color, and then it pops back up here around 650 or so. Carotenoids are yellow in color. And by the way, the colors of the line represent kind of the color of each of these. We can look here that the yellow works best somewhere in that purple range. Okay, but either way, if we look at from just a color standpoint, blues and purples and reds work best for photosynthesis. Greens and yellows do not work very well. Okay, good. So from a light standpoint, if you were going to use artificial light, you'd want to avoid green, want to avoid yellow. Purples, UV lights work really well, and red works really well for photosynthesis. All right, so we're going to talk about how these things work. So there are these pigment clusters. All right, so if we get a little closer here, so here's our chloroplast. It's got two membranes. It's got this inner membrane called thylakoids. If I look at the membrane itself, I see all the phospholipids right in here. There are these integral proteins that are stuck inside the membrane. These proteins have spots on them that actually are the pigments. Okay, here's an example of chlorophyll A, chlorophyll B. Okay, it's going to have this tail that's all hydrophobic, and it's got all these little C's, and we talked about things being polar versus non-polar. When you start looking at this, you see a lot of this is mostly non-polar. You're not seeing a lot of O's and H's. You see a lot of double bond O, double bond O. Carbon always makes non-polar bonds. Nitrogen basically makes non-polar bonds when they're stuck in a situation like this. So this is fairly non-polar. This item here. How can we relate this to the real world? If you're wearing, let's say, something nice, new and white, and you go outside and you slide in the grass and you get these pigments, you squeeze these pigments out, and they end up on your clothes. They're hard to get out with water. Coffee is derived from a bean, right? That bean is a plant-based material, has pigments in it. Those pigments, when we take the coffee and grind it up, we run hot water through it, we leach some of the color out. That's the coffee that's in our cup. If you spill that coffee on a white shirt, it stains. Why? Because it's not polar. It won't come out with just water. So kind of an interesting aside. So anyway, so these pigments here, these small dots here, these pigments are going to absorb photons or energy molecules from the sun. Photons cause your genes to fade, cause your skin to tan, cause the color of your car or your house to fade over the course of time. And we'll kind of make some analogies here about how we replace these things as time goes on, but the pigments themselves start to fade. All right. This energy, this movement of photons, is then converted from movement or kinetic energy to potential energy in the form of ATP. All right, so one of the ways we can show this is we can do phosphorescence, which is where we can take photons from the sun, along with some chlorophyll, in the presence of some other chemicals, right? And what happens is, as the photons hit the chlorophyll, they release these electrons, part of like the electron transport chain. Normally, the electrons would go right back to its own chlorophyll molecule. Kind of just it separates from it and comes right back to it, almost kind of like pulling a magnet apart and then having it stick back together. That pulling apart and sticking back together causes some kinetic energy, which can then drive the production of ATP. Excuse me. If we throw some other chemical in there, it'll absorb these electrons and it'll actually change color like this. Okay. We're going to talk about something being cyclic and non-cyclic. When I think about cyclic, I think things start and end in the same place. They make a big loop. We're also going to talk about this process of this chain reaction process where electrons, hydrogens, move from one place to another in the electron transport chain. And we'll detail this in class a little bit more. So once again, the photosynthesis has two parts. It has the light reaction and the dark reaction. The light reaction here is depicted as an up and down, same spot reaction, up and down, or it can start in one spot and bounce all over the place. These are the two types of photosystems. There are photosystems or light reaction 1 and photosystem or light reaction 2. One is cyclic, one is non-cyclic. One starts and ends in the same place, one does not. Okay. The reason that we have two of these is because plants have evolved to live in different environments. Depending on the environment they live in, it's whether they'll do one or both. So here's kind of a little diagram here. So imagine that. So we go back a couple here. So here are these small pigments, right? These pigments are bound to a protein inside the membrane, right? And here are all these little circles. This diagram here shows those pigments. This diagram shows those pigments. The number P680, P700 has to do with the color that works best in that particular pigment. We talked about earlier about how yellows and greens don't work. Reds and purples are the best. These are reds here, 680, 700. Purples would be, 500 and less. Okay, so let's talk about what's going to happen here. So what's going to happen here is that, using this diagram here as well, these pigments in plants are excited by light. Okay, so the light comes in, and the light travels as a particle called a photon. Right? We can measure them, we can catch them, we can see them. They're actually, think of it as like a proton, neutron, or electron. It's an actual small particle that light travels in. Okay. Most of the time, these photons go right through things. Sometimes when they hit our skin, we slow them down a little bit, and it causes all these different reactions in us and as well as plants. But with plants, they use the energy in these photons, and they're able to bounce these photons around. As they bounce these photons around, what happens is electrons get displaced. These electrons that get displaced are then removed from the pigment. Right? Think about this electron transport chain. Right? We talked about hydrogens and electrons separating. Right? What happens in the case of, this is the non-cyclic reaction, electrons move down a series of electron acceptors. There's a series of oxidation-reduction reactions. Right? Oxidation lost, reduction gain. Oxidation lost, reduction gain. Oxidation lost, reduction gain. And it's, I use the analogy of a hot potato. The electrons are like a hot potato. Nobody wants them. And what happens is eventually this hot potato, these electrons, are picked up by, we use the term Uber. Okay, there's this transport of electrons is what this is. Okay, same thing that happens in the mitochondria. Instead of being NAD, it's NADP. It stands for nicotinamide adenine dinucleotide phosphate, or NAD for plants. And it picks up not only the electron, but the electron, remember, had a hydrogen attached to it. The hydrogen is going to eventually, this diagram doesn't really show it well, is going to eventually follow and catch up with its electron, and when they come back together, we need to pick up these hydrogens because we don't want excess hydrogens, which lowers our pH, causes it to be more acidic. So we make this energy molecule called NADPH, which plants cannot use directly, just like we can. Imagine that if we started here and we went up and the electrons came back in the same spot, that would be cyclic, and we're going to get into that one as we go. Okay, so once again, NADPH, we use the analogy of an Uber. The idea is it's going to move hydrogens and their electrons from one place to another. It's a way of creating energy by collecting the movement of molecules across a membrane. We talked about the same thing in the mitochondria, right? We have this electron transport chain, hydrogens move through, and they use this thing called ATP synthase that drives the production of energy. The same thing happens in the chloroplast. It's just using light to drive it, as opposed to sugar or food to drive it. I'm not going to spend a lot of time on this one. We're going to go over this in class a little bit. Okay. A couple things that should look familiar here. So the Calvin cycle uses ATP and NADPH to convert CO2 into sugar. So in order to make sugar, you need energy to make sugar. Once sugar is made, the energy is stored in the bonds between the C6H12O6, or the molecules that are the elements that are found in glucose. So if you read this backwards, we could say that sugar is converted into NADP and ATP in the Krebs cycle. So it's literally the opposite of what's going on inside of respiration. So the Calvin cycle uses this ATP and NADPH can be made during the light reaction to drive the production of sugar. It really doesn't make sugar directly. What it really makes is glyceraldehyde 3-phosphate. If you go back to our conversation, glyceraldehyde 3-phosphate showed up in step 5 of glycolysis. This molecule is like Mr. Potato Head. Plants can turn this into proteins, lipids, or carbohydrates depending on what they need. And that's a whole other process that we don't necessarily get into. Just know that they're using similar molecules. All right, last part. Don't be afraid of the dark. So the dark reaction, otherwise known as the Calvin cycle. The Calvin cycle is going to take carbon dioxide, and it's going to take carbon dioxide from the atmosphere or from the water if you're an aquatic plant, and it's going to take this molecule, and it's going to turn it into sugar for us. And there are 3 phases. So there's something called carbon fixation, reductions, and regeneration. So the first part is carbon fixation. CO2 from the external environment. So it could be from air, it could be from water, depending if you're an aquatic organism. You're going to take that, you're going to absorb it into your cells, and you're going to attach it to some molecule that's already there. All right, 3 coming at a time. This is where the energy comes in. The ATP drives this process. So first part, getting the carbon into your body, adding something that's already there. Don't worry about the terms. We'll come back to these after. Second part is reduction. Remember, reduction is, so we talked about 2 things. Oil rig. Oxidation is loss of electrons. Reduction is gain of electrons. The reduction portion is the gaining of electrons. Where are these electrons coming from? NADPH is going to drop off hydrogens with their electrons. All right, and when they drop these things off, one is oxidized, NADP is oxidized, oxidation is loss its hydrogen, reduction is gain. So that's what they call this the reduction phase. So the last part of photosynthesis, now we want it to start again. So we want to bring more CO2 in, so what do we have to do? We have to regenerate or make the thing that's going to absorb the CO2, and that's the last part. It's regeneration of this carbon acceptor. So we want to make this called RUBP, or ribulose bisphosphate. We're going to remake this molecule, and in order to do that, we need energy. Notice from a total standpoint, we need 6 ATP here, 3 ATP here, and 6 NADPH. We don't have a direct accounting like we do like in respiration, where we say, oh, we made 38 ATP. What we're saying here is we're taking 6 and 3 is 9. And if you remember from before, each one of these is worth 3 ATP. So 18 and 9 would be, a total of 27. So it takes the equivalent of 27 ATP to make 1 G3P, or half of glucose. So really it takes 54 ATP just to make 1 molecule of sugar. So we only get 38 out, which kind of makes sense in the sense it takes more to make it than it's worth. We can't get 100% of the energy out of the glucose that we make. Fortunate that there's always light. All right. I mean, this time of year there's less light. When you look outside now, there's like no leaves on the trees. We know this process is not happening at a huge rate, but what's going to happen is once the weather turns, sometime around March, April, May, we start to get this huge amount of sunlight that shows up, and this process of photosynthesis will take off like crazy. All right. I'm going to skip that slide because it didn't look very good. So there are 3 types of photosynthesis. The one we're talking about is C3. That's the most common type of photosynthesis. C4 and CAM photosynthesis work exactly the same way. There's a light reaction, there's a dark reaction. Same processes take place. ATP and ADPH is being made, and at the end they're making certain types of sugars. The difference is when they take in carbon dioxide. C3 plants live in a place where it's not too warm, it's not too cold, there's plenty of water. It's kind of like the Goldilocks effect. They've got everything they need to be successful. So they can take in CO2 anytime, day or night that they want to. As long as there's light for the most part, they can do photosynthesis, so they can be taking in CO2. Plants that live in environments where there's not a lot of water or there's a lot of heat, a lot of sun, they don't like to take in CO2 during the day. And one of the reasons why is because they have these little stomata on the bottom of their leaves that they open up. And when they open those, obviously oxygen comes out and CO2 comes in. That's their way of breathing. But they also lose water. So plants that live in an area that is windy, all right, that doesn't have a lot of water, we see this technically with grasses, and plants that live in warm environments, CAM plants, they don't like to open their stomata during the day because they lose large amounts of water through those little openings in the bottom of their leaves, which they can't afford to do. So what do? They have developed or evolved a mechanism where they can open their stomata at different times of the day and at night, all right, to allow them to take in CO2 and then store the CO2 for later. So, for example, I'll use CAM plants. CAM plants only open their stomata at night. Why? Because it's colder. They don't lose water through their stomata due to the excess heat and water vapor. So they open their stomata at night, they breathe in CO2, and they store it inside the leaves, inside the body of the plant until later. You're going to record it, it's okay though.